Good afternoon, everybody. Today, my co-authors and I will provide you with an overview of the SARS-CoV-2 outbreaks in U.S. farm mink and the wildlife epidemiological investigations we participated in as part of the One Health effort resulting from those events. While humans are the primary hosts for SARS-CoV-2, there have been hundreds of spillover events into various animal species worldwide, the most significant of which are infections in farmed mink, with over 400 farms in 11 countries reporting infections. The US has reported about 59% of the confirmed positive cases in animals, with most detections occurring in dogs, cats, and farm mink, followed by zoo and other captive wildlife, and of course the one wild mink that we reported in December. With respect to farmed mink, the initial outbreaks were reported in Europe, with the most cases in Denmark, Poland, the Netherlands, Finland, Lithuania, and Greece. Europe has a much higher density of mink farms than the US, and the response in most countries there has primarily been culling of infected farms. Europe also has molecular epidemiological evidence that demonstrates transmission from humans to mink and from mink back to humans. Such reverse zoonoses, while not confirmed, may have occurred in the US. Mink production in the US is primarily through small family farms spread throughout the country. Production has been falling over the last several years, associated with a reduction in the price of pelts, but there were still 2.7 million pelts produced in the United States during 2019. Wisconsin, Utah, Idaho, and Oregon remain the top producing mink states. There have been 4 states that have experienced SARS-CoV-2 outbreaks in farmed mink. These are Wisconsin, Utah, Michigan, and Oregon. And as we saw in the previous slide, 3 of these, Wisconsin, Utah, and Oregon, are in the top 5 mink producing states. Preliminary analysis indicates that all these outbreaks are associated with human to mink transmission by infected farm workers. Biosecurity on U.S. mink farms is quite variable. Not all farms have perimeter fences around sheds to contain escapees, and those that do may or may not have features to minimize movements of wildlife on and off the properties, or to decrease the attractiveness of the farm environment to wildlife. Outbreak response of SARS-CoV-2 on U.S. mink farms has been through a coordinated One Health effort, including the CDC Special Pathogens Branch, USDA Veterinary and Wildlife Services, USGS, and state natural resources, agriculture, and public health agencies. The CDC has been sampling the farmed mink and domestic animals on the farms, as well as conducting the human epidemiological investigations with the state departments of health. USDA Wildlife Services, through the National Wildlife Disease Program and the National Wildlife Research Center, has been conducting wildlife sampling in collaboration with state departments of natural resources and agriculture. While testing of wildlife samples has been conducted by the USDA National Veterinary Services Laboratories and the USGS National Wildlife Health Center. With respect to the wildlife response, the challenge has been where to focus very limited resources, given that there was virtually no information on susceptibility of wildlife species found on and around the farm environment. On-farm investigations have focused on whether SARS-CoV-2 escaped the sheds and infected free-ranging species, and if so, was there evidence that the virus was circulating in wildlife populations? Wildlife sampling was primarily conducted using cage traps and focused on mesocarnivores. However, small mammals were also targeted on 2 of the first set of positive farms identified in Utah. We conducted wildlife sampling on 8 of the 16 positive farms in all 4 states. In response to the initial outbreak during August in Utah, all escaped mink and wildlife species captured were euthanized and necropsies were conducted to collect nasal, oral, and rectal swabs, tissue samples, and serum for testing. Results from those initial investigations indicated that all confirmed detections were identified through PCR testing of swabs or virus neutralization tests on serum. Therefore, in subsequent outbreaks in Utah, Michigan, Wisconsin, and Oregon, animals were euthanized and only swab and serum samples were collected. Today, we'll share with you the preliminary highlights of the wildlife sampling conducted in all 4 states. Across all farms, we captured just over 250 mammals, including mesocarnivores, rodents, and lagomorphs. Of those, we only confirmed positive detections in mink. We captured 21 mink, 17 of which were escaped farmed mink in Utah and Oregon, and 4 were wild mink in Utah and Wisconsin. Note that we did not capture any mink associated with the single positive farm in Michigan. Of the 17 escaped mink, 16 were positive by PCR, serology, or both. Only 1 of the wild mink was positive, and it was positive by PCR. We also recovered a virus from that individual. While sequence analyses are in progress, preliminary results indicate that all 3 viruses we recovered from escaped and wild mink are identical to those found in the farmed mink. Now, as we indicated in our OIE report in the ProMed Mail post on December 12th, the wild mink we found positive was the first known case of a free-ranging native species confirmed with SARS-CoV-2. That wild mink was captured in a natural creek habitat 672 meters from an infected shed in Utah. In this diagram, the red dot labeled A is the infected shed, and the purple dot labeled C is the capture location of the wild mink. The green dot labeled B is the location of an open pit where carcasses of dead infected farmed mink were being deposited by the owner. That pit was about 70 meters from the creek and only 242 meters from the capture site. The yellow dot represents the point at which the creek is closest to the sheds, about 578 meters. Given that the mean home range size of a wild mink is about 5 square kilometers, the mink could have been exposed to SARS-CoV-2 by accessing the farm site directly from the creek or by contact with infected carcasses in the pit. An interesting side note to this story is the challenge we had in determining whether the mink we captured were wild or recently escaped farm mink. Given that farm mink are the same species, Neovison vison, distinguishing the two is not straightforward. Genetic analyses of individuals to classify the animals as escaped versus wild was of limited value because an adequate genetic reference library for wild mink in the US does not currently exist. Also, studies in Canada, such as this recent one by Bowman et al., found that farmed mink regularly escape and that escapees can hybridize with wild mink, complicating wild versus farmed identification based solely on genetics. However, we were able to conduct microsatellite analyses to determine if a captured mink was genetically similar or distinct from farmed mink sampled in the area. We identified 8 genetic clusters, many shared between the single farm in Oregon on the top and the 4 farms in Utah on the bottom. Here, each rectangle represents a sample for an individual mink. The colors represent genetic clusters, and the letters represent the 4 farms in Utah. The wild mink that was positive for SARS-CoV-2 is represented by the dark green rectangle at the bottom right in this diagram. Notice that it does not cluster with any of the farmed mink samples tested. Also notice that the 2 dark brown rectangles just to the left of the dark green one represents our 2 other wildlife mink samples from Utah. While these 2 wild mink samples clustered with 1 sample from a mink in Farm B, we still call these mink wild. That's because given the limited value of genetic analyses and the propensity for some escaped mink to hybridize with wild mink, we relied more heavily on other characteristics in deciding if a captured mink was wild or escaped. For example, was the animal caught on or off the farm? Was it in native habitat or in a farmed or other human-altered habitat? Was it morphologically similar to farmed or wild mink? Wild mink are typically milk chocolate brown, whereas farmed mink are black, white, or silver. Also, wild mink have prominent guard hairs, as you can see on the top animal here, while farm mink have few guard hairs, such as the black animal on the bottom. Additionally, live-trapped escaped mink tend to exhibit relatively docile behavior as opposed to wild mink, which are aggressive in traps. And if necropsies are performed, physiological differences can sometimes be observed, such as the presence of large amounts of subcutaneous fat and even fatty livers in escaped mink, which are rarely seen in wild mink. Given the relatively focused trapping effort on and within a 3-kilometer buffer of infected farms, evidence to date suggests that although the SARS-CoV-2 virus escaped the sheds on some of the infected mink farms, there is currently no evidence that the virus has become established in wildlife populations. Furthermore, given that the virus is identified in farmed, escaped, and the wild mink were identical, it is unlikely that the one isolated from the wild mink passed through an intermediate host of another species. This suggests that the wild mink likely contracted the virus either through direct contact with an infected farmed or escaped mink, or through indirect contact with infectious material such as carcasses, feces, or other attractants. It's also important to remember that currently the SARS-CoV-2 virus is a human-adapted pathogen. We know that SARS-CoV-2 infection spreads most commonly from person to person. Infected people, either with or without symptoms, can introduce the virus to farmed mink. Once introduced into a shed, infected mink rapidly spread the virus to other mink, resulting in mortality rates from essentially 0 to over 40%. We're still learning about how long mink are infected with the virus and how it can spread to other animals on the farm, like dogs, cats, escaped mink, and wildlife. And finally, at this time, there's no evidence to suggest mink to human transmission has occurred on US farms, as was observed in Europe. State Departments of Agriculture and Health, the CDC, and USDA are working with farmers to increase awareness of the potential for workers to infect mink, to heighten the biosecurity efforts on farms, and to develop active surveillance plans for the upcoming breeding season. While currently there are no actively infected farms in the US, if additional infections are identified, wildlife surveillance will be included as part of the One Health investigations. Also, experimental infection studies are being conducted to determine which species are at greatest risk of infection. A recently posted preprint paper by Colorado State University and the National Wildlife Research Center found that deer mice, bushy-tailed wood rats, and striped skunks were susceptible to experimental infection with SARS-CoV-2 and shed the virus in their respiratory secretions. Alternatively, cottontail rabbits, fox squirrels, Wyoming ground squirrels, black-tailed prairie dogs, house mice, and raccoons were not susceptible to infection. Finally, we wish to thank all our state and federal partners for their participation in these One Health investigations, especially to those landowners who gave us permission to trap on their properties. Thank you very much.